Hello everyone. Today we are going to be talking about E. coli and its specific serotype of 0157H7. The two objectives I want to kind of focus in on is what is E. coli. I don't want to look just at this whole presentation is going to be looking at the specific serotype, and I want to explain what E. coli is and how we get to that serotype and how it's different, its differentiation between other E. coli, and also going into how does it affect you, where you might see it in production, how does it get there, where you might see it in other places. So you'll be able to see E. coli is not just in beef, but you can see it, or not even just in livestock. You can see it in vegetables. You can see it in also in things such as ciders, apple ciders. And I kind of want to get onto that whole aspect of how it might affect you and what to look out for. All right, this is a picture just to give you an idea of what E. coli looks like. It is a rod-shaped gram-negative, facultatively anaerobic bacterium that normally populates the GI tract, and in this case, or in all cases, it will normally populate the lower ends of the GI tract. This is because as you go down the GI tract, you'll start getting a higher pH, and it prefers to be in a higher pH compared to the duodenum. The duodenum normally keeps a pH around 4. Whenever you get down farther down the GI tract, you'll see it increasing pHs of about 6 to 7.4. And especially in livestock, it normally likes to populate the renal anal junction. It is very popular, and not in a good way, for causing urinary tract infections or hemolytic uremic syndrome. And it is probably world known to cause bloody diarrhea, as a lot of people like to see it as. In order to give you an idea of the environment that E. coli likes to be in, it did evolve and usually in warm mammalian GI tracts. So if you think about just your own GI tract, you can kind of get a good idea of what environments it likes. So I've already told you about the pH that it prefers. It also prefers a 37 degrees Celsius temperature, which is 98.7 degrees Fahrenheit, which, as we all know, is our normal body temperature. So right here, I want to talk a little bit more about serotypes. A lot of times when you hear E. coli 0157H7, which is the most popular one, everyone kind of puts this into as a broad umbrella over all E. coli, which is not the case because a lot of the times E. coli can actually be good for gut health, but not the serotype 0157H7. This is just going to be telling you why different E. coli can be good and why other ones can be bad. So in this case, serotype helps us subdivide from different E. coli. This is due to surface antigens that are found on lipopolysaccharides on the outer membrane that attach these things called O-specific polysaccharide chains, which I have labeled above. So these O-specific polysaccharide chains are what allow us to divide and determine between different serotypes of E. coli. They also help us bind these E. coli to endothelial cells. So going off of what I said in the previous slide, I was talking about how these pathogens are able to bind to endothelial cells, but going on to the next step, they're able to produce these things called Shiga toxins. Shiga toxins are actually going to be targeting these things called GB3. GB3 is a globicide. A globicide that is formed by alpha linkages of galactose and lactosylceramide. Sorry, it's hard to pronounce that one. And actually is able, once it's able to target these GB3, it allows and aids in the pathogenicity. So without these GB3s, there wouldn't be any expression or any of these symptoms of UTI or bloody diarrhea. If we actually had a deficiency in GB3, we had no expression of GB3 whatsoever, that actually causes something called Fabry disease, which I won't get too in detail into it because that's just kind of a side note. The reason why I wanted to talk about this, and the whole reason why I want to talk about serotypes altogether, is because it's important to know that cattle actually lack the expression of vascular GB3. This is important to know because without this expression of GB3, Shiga toxins aren't able to be absorbed into the bloodstream and therefore distributed to different organs. So this is the main reason why we express these symptoms when we have the serotype 0157H7 and cattle don't kind of give off or kind of express the same symptoms we do. So right here is a table I took out of one of Dr. Robert Elder's publications. He is a researcher at the United States Meat Animal Research Center. This was a publication done around the year 2000, specifically on January 19th is when this publication was made. It was simply a survey done on, as you can see, 4 different plants, and then they're subdivided into 2 samples, and these samples being further subdivided into 3 to 4 lots per sample. This was kind of giving you an idea of where you might see E. coli being transferred. We see it in the feces more than anything. It's being shed into the feces or the hide. It also gives you an idea of where you might see it on a processing side or at what step you might see it the most prevalent. So in this case, we see pre-visceration, post-visceration, and post-processing. Pre-visceration being before the viscera is removed, so something such as organs. And we can see it kind of goes from a higher to lower, so this kind of makes sense. So as it goes through and proper sanitation procedures are done, you'll see this E. coli being, and then also the environment isn't as optimal for the E. coli to survive. You can kind of see it deteriorating of how much is prevalent, but you can still see a little bit still survive into the post-processing. This project was also done in July through August. July through August is summer to early fall, so this is the time you'll mostly see E. coli more prevalent. This is probably due to the fact that during those times it has a more optimal temperature, so when it's getting shed into the feces, it can survive longer in that feces during more optimal temperatures. This being, again, like I said in previous slides, around 98 degrees Fahrenheit. So during those late summers or August, especially in Georgia, you can definitely see the temperature getting up that high. I apologize for the slide being kind of cluttered, but I wanted to keep everything here because it all kind of ties together. The first table above was taken out of a paper published by Dr. Tong Zhao of the University of Georgia. It was published in 1993 and kind of gives us an idea of the prevalence of E. coli in apple cider. Apple cider, unpasteurized apple cider normally keeps a pH around 3.6 to 4, and when refrigerated, it will keep around a temperature of 8 degrees Celsius. At least in this project, this is what they were going off of. This was done on 6 different lots of unpasteurized cider. The first one being the apple cider only. Then we're looking at 2 preservatives known as potassium sorbate and sodium benzoate, and a combination of both those preservatives as the 4th test. The results you can kind of see right here that when apple cider is left to its own, that the pathogen still kind of dies off and is not as prevalent. This is due to the fact of that 8 degrees Celsius environment and the fact that, again, apple cider is normally keeping around a pH of 3.6 to 4. Both of these preservatives, the potassium sorbate and sodium benzoate, were effective, but potassium sorbate didn't show a significance when compared to apple cider. But sodium benzoate and a combination of both the preservatives did show significance when compared to just the apple cider with no preservatives whatsoever. This kind of gives us an idea that why preservatives are used. These preservatives themselves actually was stated that its ability is able to lower the pH of the cider, so by 0.1 all the way up to 0.4 pH to lower it by that much. So this is creating that environment where it can't, it's not optimal for its growth. So that's probably why you see a slight decrease in potassium sorbate. It's not as effective compared to sodium benzoate, which is able to decrease pH further and kill off more of that pathogen. Going on to the next 2 tables, I kind of wanted to add these in because if people are still curious about how we get from feces and then beef in general and how we get to these infections in these vegetables and fruit, the reason is because a lot of our soil is probably manure based that we get off of different plants with the feces. So these 2 tables were taken from a professor known as Ethan Solomon of Rutgers University. It was published in 2002. They used a 5 kilogram soil sample, a mixture of 4.5 kilograms of soil, and that remaining 0.5 kilograms was manure infected with different concentrations. You can see there different concentrations of the pathogen. They wanted to test the ability of a lettuce seed and see whether or not, when left to its growth in the infected soil, if that E. coli would be able to penetrate inside the seed and not just infect the outer surface. As you can see from the results, there is a mixture of inner infection and outer infection. But when further subdivided and looked at in more detail, they found that even though some were found on the outer surface of the seed, that it was on the subsurface. So it was still going into the actual seed in itself. This is important to note because a lot of times when lettuce is harvested, it's washed down with certain chlorinated water. But if this pathogen is able to survive inside, a lot of this surface cleaning won't be as effective as what they think. Kind of going off of that, we look at table 2. This is simply just a table to kind of give you an idea of how contaminated irrigation of water that you might see being applied to the plant itself can further populate and penetrate into the actual inner portion of the lettuce. This being because when they planted these seeds in this contaminated irrigation water or the contaminated manure slurry, they wrapped the lettuce in actual twine. This prevented the surface of the lettuce from touching the soil, and they wanted to see if this infected irrigation water or the contaminated manure slurry would actually take root, and then by that root go into the lettuce and then spread out into actual edible parts of the lettuce. So when tested, they took 5 of the lettuce plants, and they found that 4 out of the 5 on the first day were infected, 2 out of the 5, and then also 2 out of 5 on the 5th day. When they looked at contaminated manure slurry, they noticed there was still just as much that were still infected. This can kind of give you an idea of where you might see runoff being a problem for that contaminated manure slurry and so on and so forth. So this kind of gives you an idea of just because if you maybe a vegetable or fruit is washed thoroughly, this pathogen can still be prevalent within that vegetable or fruit. So in conclusion, we talked about E. coli, kind of what the importance of knowing its serotype and how it might be different from other E. coli, that there's not always just a bad serotype like the 0157H7, but there's also can be good E. coli, what serotype is overall. We went into beef and dairy a little bit of where we see it in both those industries, and also aside from the livestock, but still kind of connected, we talked about its prevalence in lettuce and apple cider. It's important to kind of know these things, but it seems like it would kind of worry you for a lot of the presentation I gave. But to be honest, that only 0.00002% of the population, I think it was 4 or 5 zeros, percent of the population actually die each year from E. coli or the E. coli serotype 0157H7 infections. It can be life-threatening, but it's not something that you need to be completely worried about. But it's also really, really good to kind of know these past projects to kind of give you an idea of where research is going. And just to give you an idea of how we've studied it enough and how there's a reason why it's so low. I'm sure a lot of processors and production operations look at these papers and kind of get an idea and able to improve their sanitation operations and protocols. So this research is very important. It's very good that we research this early because a lot of these papers were done in the early 2000s and later 1990s. So that it's probably the reason why we don't see a lot of prevalence in E. coli today, or why we have that 0.00002% of deaths per year. It's just because we've done really well in researching and knowing how to combat this pathogen. So that's it for this presentation. I want to put some links below where I found a lot of these things if you want to research it yourself. And yeah.